Um, I trust that this is your evening class and you're going to be looking further into DNA. But from the morning class, a couple of you have missed it, so I'm gonna have to start from the beginning, so from the top. Firstly, looking at the revision of the animal cell. Okay. Now, of course, there are also plant cells, which is quite a little bit similar to the animal cell. The only difference is that the shape of a plant cell is irregular, so it can be like all wobbly and stuff without a proper structure other than a animal cell. And also a plant cell has a component called the chloroplast. In animal cells we do not have the chloroplast. Alright, but anyways, what is more important right now is to go through the animal cell in detail. Alright, okay, so now looking at the animal cell, it is regular shaped, and the outside membrane here in purple, in this purplish color, is the cell membrane, and then inside there is this jelly-like fluid, which is in blue, is the cytoplasm. Now this jelly-like fluid has the function of maintaining the shape of the cell, and also, since it's a fluid, it's to also prevent the cell from dehydrating. Okay, and now moving on into the cell, there are other organelles that we will not look into in detail in our syllabus, but it will also help to keep those in mind. But then the most useful ones are the ribosome, which are those dots that you see in purple inside the cytoplasm. Now the ribosome is the site where protein synthesis takes place. We will look into protein synthesis in detail soon, and you're gonna need the nucleus and the mitochondrion. Okay, now mitochondrions all together, we call them mitochondria, but when we are referring to one mitochondria, we're going to call it mitochondria. When it's many of them, it's mitochondrion. Okay, now mitochondrion provides power for the cell, and then the nucleus is where our DNA will be found. Now DNA stands for deoxyribonucleic acid. Okay, now our deoxyribonucleic acid contains the genetic code for all living organisms. So it's basically what differentiates between different organisms, if I can say. So when we look at DNA, it is actually in the nucleus. There are also other places in the cell where you can find the DNA, but the one we're going to concentrate on is the DNA found inside the nucleus. And the DNA found inside the nucleus, we call that nuclear DNA. Okay, so now looking inside the nucleus, there are these thread-like structures inside the nucleus called the chromatin network. Now inside those chromatin network, we have things called chromosomes. Now those chromosomes are what has the DNA, which we're going to be discussing shortly. Alright, now this DNA it basically differs from organism to organism, from person to person. Okay, so now looking at this animal cell, let's say per se the DNA that forms your skin. Let's say for instance you get injured and part of your skin peels off. Your skin needs to find a way of healing itself and creating new, if I can say, overlapping the gap with new skin that looks exactly like the one that has peeled off. So now it has to create a copy of a cell, the cell that was there, and create a copy that is exactly the same as that cell, so that it can form part of that skin. Now for it to be the same, it means that the DNA in the old cell has to be similar as the DNA in the new cell. Now the process of creating new cells that are exactly identical, we know this process as mitosis. Okay, now mitosis is basically when you have one cell that divides into two cells that are exactly the same. Okay, now these cells are genetically identical and this will only have one division. It will only divide once. Okay, now we have another process called meiosis, kind of similar to mitosis but occurs at two different places. Mitosis takes place in somatic cells. Somatic cells are body cells, but in grade 12 you will not refer to them as body cells, but you will refer to them as somatic cells. So now mitosis is somatic cells, meiosis is going to be what we call gametes, and gametes are sex cells. So in males we'll be talking sperm cells, in females we'll be talking ovums. Okay, or you may refer to them as egg cells. Okay, so now in meiosis we're not only going to have one division, we're going to have two divisions. In mitosis we're going to have two divisions. In meiosis the cells that are going to be formed are going to be four cells. In mitosis you're just going to have one cell that divides into two cells. Now those two cells that are going to be formed are going to be genetically identical. Now in meiosis they're going to form four cells. Now these four cells are going to be genetically unidentical. Now the DNA inside that nucleus is going to be unidentical in all four daughter cells that are formed. Okay, so now we're going to go into the process of DNA replication. So I've prepared a slide here about DNA replication, right? So DNA will need to make copies of itself during the phases of mitosis and meiosis, which we will look more into detail in our next lessons. So now moving on, what is DNA replication? All right, so during mitosis, we've spoken a little bit more about mitosis. It's where we're creating two daughter cells that are genetically identical. Okay, so if you had your old skin, it should look exactly the same as the new skin. Okay, so that's basically what genetically identical means. Okay, then the two daughter cells that are formed should be genetically identical to the mother cell. So you're going to have one cell that is going to divide itself into two cells that are exactly the same. So if this cell was to be called A, this cell should be called A and A. They should be genetically identical to one another and also genetically identical to the mother cell. Okay, so that's what we mean. So this means that the DNA needs to make copies of itself that are identical, which means the DNA in this should be the same as the DNA in that and the DNA in that. Okay, so the process of duplicating the DNA is known as DNA replication. You might find this question as part of the biological terms of your exam. Okay, now the DNA replication takes place in the nucleus of the cell. I'm sure I've mentioned where the DNA is in the nucleus of the cell. Okay, so inside your cell you have your cytoplasm, which is the jelly-like fluid which is all around there, and then you have the nuclear membrane. Okay, so now that nuclear membrane is kind of like another cell as well, but now the nuclear membrane has holes. Okay, so I'm just gonna put it like that. So it has kind of like holes right there, and we call these holes nuclear pores. Right, now inside that nucleus there's another jelly-like fluid as well. Okay, now this jelly-like fluid is called the nucleoplasm. Okay, so that's a jelly-like fluid in the nucleoplasm. Then we have a thread-like structure which is kind of like this, which we call the chromatin network. Now inside this chromatin network are what we call chromosomes. Now chromosomes are kind of in an X-like shape like this. Okay, so they have three parts to it. We have this part right here and this part. Okay, so let me complete the drawing, and we have something right here in the middle, so I'm gonna label it now. Alright, so now this right here is a chromatid, and that's where the DNA is. and this here is another chromatid with DNA as well. Now what holds these two chromatids together is what we call a centromere. Okay, I'll send you detailed drawings later on. What holds them together is a centromere. Now this chromatid here and this chromatid here have DNA in them. Now a certain segment of a chromatid, kind of imagine this as a step ladder and a line going this way. Kind of imagine that each segment codes for a specific characteristic, and each segment we call it a gene. So for example, we have a segment that codes for the hair color, so that is a gene for hair color. I hope that makes more sense. Alright, so now moving on, I will erase these. Now moving on. Right, the process of DNA replication. Now I'm gonna explain this in a condensed package. I'm gonna try to redraw it. First step is the unwinding of the DNA double helix. Now DNA has a specific structure in nature called a double helix. This means that if it is not winded up, it is kind of in a step ladder in this way. Okay, it's kind of in a step ladder. Then when it is winded up, it goes into a structure called a double helix, which means it will be something like this, Alright, so now our first step of DNA replication, this double helix has to unwind into the ladder. Okay, so now we do know, I'm quite sure you do know the shape in nature of a DNA molecule. A DNA molecule, DNA is made up of nucleotides. Nucleotides are building blocks of DNA. You might need to write that down. Now this is how a DNA nucleotide is formed, has a structure. So it is kind of in an L shape like this. Alright, so it's kind of in an L shaped. So right here we have phosphorus, okay, and then here we have deoxyribose sugar, and then here we have a nitrogenous base. Okay, so this is the basic structure of how it looks. So what's going to be the same each time is basically the phosphorus and the deoxyribose, but now the nitrogenous bases, there are different types of nitrogenous bases. There are actually 4 types of nitrogenous bases. Alright, and namely these are adenine, guanine, thymine, and cytosine. Okay, so those are the types of nitrogenous bases we have in DNA. In RNA we have 4 nitrogenous bases as well, which is adenine, guanine, and cytosine and uracil. The only difference is that in DNA you've got thymine, and in RNA you've got uracil. Now looking over at the nitrogenous bases in the structure of the DNA. Now keep in mind it's the phosphorus and the deoxyribose sugar. Now the entire line here going down. So if you look at this in an entire DNA molecule, from here you're going to join to another deoxyribose sugar, which is bonded to another nitrogenous base, and again, do you see this? A phosphate, and keeping on and keeping on. So this keeps going, and you keep on having an entire strand forming. So one piece of this is called a nucleotide, and then when you have nucleotides together, that's when you have a DNA strand. Now keep in mind that a DNA is double-stranded, which means if you have one side, you need to have another side. So you're gonna have another side like this, another phosphate, another deoxyribose, and another nitrogenous base. I mean. So now our 4 nitrogenous bases have certain ways in which they bond. We call those complementary bases. All right. So looking at them, we have our nitrogenous bases, we have adenine, we have guanine, we have cytosine, and we have thymine. Now there are ways in which they have to bond. Alright, so now there is a pseudonym that I like to tell people to help you remember the ways in which they bond. Okay, cute girls all taken. This will help you remember how they bond. C with G, A with T. Cute girls all taken. It is that easy. Now C with G would be cytosine with guanine, and A with T would be adenine with thymine. So if you look at these nitrogenous bases, if you had adenine here, if you look at this, A goes with T. So if you had adenine there, which means you'd have T there, you would have to have thymine on the other side. If you had thymine there, you would have to have adenine on the other side. If you had cytosine, you would place guanine on the other side. Now there is a bond between these nitrogenous bases. Right here there is a bond that is supposed to be placed here, and the name of this bond is called weak hydrogen bonds. Alright, so the bond between the nitrogenous bases is weak hydrogen bonds. Alright, now back to the DNA replication. Now let me quickly get my laser pointer right there. So our DNA double helix, as we said, unwinds. So its shape in nature is kind of like this. So when it has unwinded, it's going to become like this. Alright, so we do remember our nitrogenous bases are somewhere around there. Alright, now the next step, which is step 2 here, these nitrogenous bases have to be broken. Okay, so this is the unzipping process where the weak hydrogen bonds are broken. So which means the bonds between here are going to be broken. Okay, so one goes each side, the other goes the other side. Okay, I hope that makes more sense. So they're going to unzip. Okay, and then one strand will serve as a template, which means, for instance, this strand will serve as a template for our DNA replication process. Okay, so I'm going to use this template. I'm going to use that one as a template. So it's kind of like that. Okay, now remember this is taking place inside the nucleus of the cell. Okay, now inside that nucleus there are free floating nucleotides. Okay, in that nucleoplasm, the nucleoplasm is that jelly like fluid we spoke about inside the nucleus. Now there are free floating nucleotides. If you remember what nucleotides are, it's that sugar phosphate and nitrogenous base thing we were talking about. So these things are inside the nucleoplasm. They are free floating. Okay, now remember the ways in which they complement: cytosine with guanine, adenine with thymine. Okay, now keep in mind with that. So now these free floating nucleotides are going to go ahead and complement the triplet bases on that template strand. So for instance, here if we had cytosine, if we had adenine, and here we had guanine, so the free floating nucleotides that will complement this is that we'll have one that has guanine here, which will complement with cytosine, and here we'd have one with thymine that would come here, and here we'd have one with cytosine that would come here, and then before you know it, you have an entire DNA molecule. Now the other strand as well, the same process would happen. Now notice this, the other strand here, if we can redraw its coding, cytosine, the other strand here would have guanine. I'm talking about this strand here, would have guanine, and the other one would have thymine, and the other one would have cytosine. So if you see the nitrogenous bases that are going to come and complement this, is that one would have cytosine, one will have adenine, and the other one will have guanine. Now before you know it, these two strands are exactly the same with the pattern of nitrogenous bases. This means that these two strands are now genetically identical. Okay, I hope that makes more sense. Alright, so I'm going to go ahead and erase this and show you the motion picture in the background. Alright, here. Now if you look at this motion picture right there, this GIF, these are the free floating nucleotides you would have inside the nucleoplasm, and then here you've got one template cell in another template cell, and then here at this point there are two processes taking place, the unwinding and the unzipping. Okay, so now here you're having the free floating nucleotides being placed onto the template strands, and then this strand and this strand would be genetically identical. It is that simple. Now that's DNA replication in a nutshell. Alright, now the significance of DNA replication. Alright, DNA replication takes place during interphase of mitosis. Now I'm not quite sure if you remember the entire process of mitosis, but it's not going to be examined, but it's quite important that you remember this point. Alright, now the processes of mitosis, if you remember them as IPMAT: interphase, prophase, metaphase, anaphase, and telophase. Now interphase is where DNA replication takes place. Alright, so now if this was you in grade 10 or 11, you would remember this interphase, prophase, metaphase, anaphase, telophase as I promised Mike a tablet. But then it is okay, you will not be assessed on the processes of mitosis, but you will be assessed on meiosis. Alright, so I'm gonna go ahead and erase these. Right there. Alright, so now during mitosis, remember I gave you the example of your skin healing itself. So now this DNA replication ensures that the genetic material formed to make up each cell is exactly the same. Okay, and it also makes sure that there is enough. So imagine that if after DNA replication only one is formed instead of two, there wouldn't be enough genetic material to form the new cell. Okay, so that is the significance of DNA replication. Moving on to our next chapter, which is protein synthesis. I hope you're able to grasp these as much as possible. Now, protein synthesis. Protein synthesis is the manufacturing of protein molecules from a DNA strand. Alright, so we remember the parts of the cell, we remember where the DNA is. The DNA is inside the nucleus. Okay, so now protein synthesis takes place in two steps: transcription and translation. We'll go through those. Now, protein synthesis begins in the nucleus of the cell, where the DNA is. Remember, we need to begin with the DNA strand first. So this takes place beginning in the nucleus of the cell. Alright, and this entire process ends on the ribosome outside the nucleus. Alright, and the ribosome is inside the cytoplasm. Alright, so if we had our cell, our nucleus, now this is where those nucleopores, those holes on the nuclear membrane I spoke about are. So now we're going to have our DNA somewhere in there, and then we're going to have something that needs to leave and go to the ribosome in the cytoplasm. Alright, now we're going to discuss that in detail in a bit. Alright, so now just remember the names of the two processes that take place. It's transcription and translation. Now transcription goes first and translation is last. Alright, now transcription is the first step. Alright, so now it's kind of similar to DNA replication, but now the difference is that the strand that we are forming is not a new DNA strand, but it's an mRNA strand. Now if you remember what RNA is, RNA stands for ribonucleic acid. Okay, so now the difference between DNA and RNA is that DNA is double-stranded and RNA is single-stranded. What we mean by that is single-stranded is that it only has one side. Alright, so we have our nucleotides, RNA nucleotides, and that's it. There is no other side to this. Now with DNA, it's double-stranded. You have one side to it and you have another side to it. Right, this is what we mean is double-stranded. There are weak hydrogen bonds here, but with this there is no need for nitrogenous weak hydrogen bonds, I mean. Alright, now with RNA we have uracil. Alright, uracil. we have uracil, and then with DNA we have thymine. Thymine. Right. Now, if you remember the nitrogenous bases in these ones, in DNA that we mentioned, adenine, guanine, cytosine, and thymine. So if you remember them, that's cute girls all taken. Now with RNA, instead of thymine, they now have uracil, and all the other nitrogenous bases remain the same. So it's no longer cute girls all taken, but it's now cute girls all used. Okay, so something like that. So we know that it is double-stranded, it has uracil, and another thing, if you remember the structure of a DNA strand, is that, or rather not a DNA strand, but a DNA nucleotide, is that you have a phosphorus, you have a deoxyribose sugar, and you have a nitrogenous base. If you remember this nucleotide we discussed before, a phosphorus, a deoxyribose sugar, and a nitrogenous base. Now the difference with RNA is here. Instead of a deoxyribose sugar, it is a ribose sugar. Not a deoxyribose sugar, but a ribose sugar. Okay, so this one has a ribose sugar, okay, while this one has a deoxyribose sugar. So that is their differences. All right, now looking more into RNA, all right, we have types of RNA, and with DNA we have types of DNA as well. Okay, now the types of RNA we have is mRNA, okay, and another type of RNA is tRNA, okay, and the other one we have is ribosomal RNA. Okay, all right, RNA. Okay, then DNA, we have nuclear DNA, which I mentioned. All right, and then we also have DNA found inside the chloroplast in plants only. We have chloroplastic DNA. All right, and then we have what we call mitochondrial DNA. This is DNA found in the mitochondrion. Okay, now this mitochondrial DNA is only going to be, we're only going to discuss it when we're doing evolution. So we don't have much significance in discussing this right now. All right, so now you might need to jot those down. In RNA we have mRNA, which stands for messenger RNA, and then in tRNA stands for transfer RNA, and then we have rRNA, which is ribosomal RNA, and then in DNA we have nuclear DNA, chloroplastic DNA, and mitochondrial DNA. All right, now mRNA is found in the nucleus, tRNA is found in the cytoplasm, and rRNA is found on the ribosome. All right, now I'll clean that out. So now back to transcription. So now transcription, we said protein synthesis first takes place inside the nucleus. All right, inside the nucleus. So our DNA double helix unwinds and unzips. I'm sure you quite remember the process of DNA replication we just discussed just now. So it's going to unwind and unzip. All right, then our free floating nucleotides, all right, these are no longer DNA nucleotides, these are now RNA nucleotides. All right, so this thing has unwinded and unzipped, so it's something like that. So our free floating nucleotides are going to go ahead and complement. They're not going to bond to this, but they're just going to complement, sort of follow the pattern. So if this was C, A, and T, so now in tRNA, C would complement with G, A would not complement with T, but will complement with U, because we're talking about RNA here. Okay, so that would be U and no longer T. Now since this is T, then it would be A this side, because it's a tRNA strand. All right, now these two will not bond. There's not going to be a bond between these two, because this thing will need to escape the nucleus and go to the ribosome. Okay, so now this strand that forms is now our mRNA strand. That's our messenger RNA strand that forms there. Okay, so now remember, this is still inside the nucleus. Okay, now remember our nucleopores, the holes between the holes in the nuclear membrane. So now our mRNA strand is there. So our mRNA strand is now going to escape through the nucleopores into the cytoplasm. Now as it goes into the cytoplasm, it will go to the ribosome, and that will be the next step we're going to discuss, which is translation. Alright, now translation. Now our mRNA strand is going to go ahead and attach to the ribosome. Okay, then there are now free floating tRNA molecules that are carrying amino acids. Now let's discuss proteins a little bit more. Remember, we're trying to create proteins here. Okay, we're going to create proteins. Proteins are built up of amino acids. These are the building blocks of proteins. Now amino acids are now found in the cytoplasm, carried by tRNA strands. Now let's take a look at this motion picture. Alright, this is a tRNA strand. Alright, it has an amino acid. It's carrying an amino acid. Now remember the nitrogenous bases. A tRNA strand has 3 bases right there. I'm sure you're able to see that. Now those 3 bases on a tRNA strand, let me try to redraw it. So our tRNA strand will be something like this. Okay, it's going to be something like this with legs of some sort. Now on the feet, it has nitrogenous bases right here. There are 3 nitrogenous bases here. Let's say, for instance, we could have cytosine, adenine, and guanine here. So now these 3 bases, as long as you have 3 bases and they are on a tRNA strand, we're going to call them anticodons. Okay, and then there are 3 bases on the mRNA strand, which is somewhere there on the mRNA strand. Remember, the mRNA strand is on the ribosome at this point. Now on that ribosome, there are 3 bases. Okay, so it's going to be something like this. On the mRNA strand, it's going to have 3 bases on it. Right, remember, it's attached to the ribosome. Okay, so now right there are 3 bases, and those triplet bases on that mRNA strand, we call those codons. Okay, we call those codons. So now these triplet bases are going to go ahead and complement these here. Remember, this thing is carrying a specific amino acid. Okay, so it's carrying a specific amino acid. It's going to bring it to the mRNA, and then at the same time, there's another tRNA that's carrying another amino acid, another tRNA strand carrying an amino acid. It has its own anticodons. Okay, it's going to go ahead and complement the next 3 bases, and remember, these amino acids are going to be brought up. Okay, so now let me clear this and take you back to the motion picture. So triplet bases, triplet bases, triplet bases. Now you need to look here. These amino acids are going to be left on the mRNA strand. They are going to be left there. Okay, now those tRNA strands are going to go back into the cytoplasm and bring back more amino acids. Okay, they're going to keep on bringing more amino acids in this sequence that you see right there. Okay, now the sequence in which these acids are going to be brought up will give you your protein. Now the way in which these amino acids are placed together is in a chain. Now this chain has a bond between them. So these are amino acids. Now there's going to be a bond between them. Okay, there's a bond between the amino acids, and the name of this bond is called a peptide bond. Okay, it's called a peptide bond, and then this entire chain is called a polypeptide chain. Okay, so this bond is a peptide bond, and the entire chain of amino acids is a polypeptide chain. Now the specific shape in which all these amino acids are in will give you your protein, and that's basically protein synthesis in a nutshell. Okay, so now an overview and summary of the 2 things we've discussed: protein synthesis and DNA replication. DNA replication, okay, is basically making 2 identical copies to the original double strand. Okay, and this takes place in the nucleus, And the processes was unwinding, unzipping, one serving as a template, the free-floating nucleotides complementing the bases on the template strand, alright, and two strands will be formed that are exactly identical, alright, and then this takes place during interphase of mitosis, right. The protein synthesis is the manufacturing of proteins. It takes place in the nucleus and at the ribosome, and the two steps were transcription and translation. Transcription, remember, is basically the unwinding, unzipping, and the formation of the mRNA strand. Then the mRNA strand has to leave the nucleus through the nucleopause and go and attach to the ribosome. And then translation is where we had the tRNA strands rather, bringing the amino acids to the mRNA strands where they are codons and anticodons. Now the order of amino acids describes the shape and form of the protein that is formed. Now the chain of amino acids is called the polypeptide chain, and the bond between each amino acid is the polypeptide bond. And I'm sure you remember that. Now that's basically DNA replication in protein synthesis in a nutshell, and that ends the entire class today. Thanks for watching, and from this you will get a quiz. You will receive a quiz based on everything I've gone through, and I hope you'll do well in the entire quiz, and I'll see you in the next one.